Somebody's going to tell you it was the turkey, that there's some chemical hiding in the meat that explains why you're basically fighting to keep your eyes open 20 minutes after a big meal. This story has been passed around dinner tables for decades. It's practically a holiday tradition at this point. It's also almost entirely wrong. Turkey does have tryptophan in it, sure, about 350 milligrams per 100 grams. But so does chicken. So does cheddar cheese, actually in a higher amount. And nobody's ever blamed their afternoon slump on a grilled cheese sandwich. The real reason your body shuts down after eating isn't about one ingredient at all. It's at least 3 separate biological systems, each running on its own clock, that just happen to slam into each other in your body every single afternoon, and only one of them actually cares what was on your plate. Let's start with the one that has nothing to do with food. Back in 1996, a sleep researcher named Timothy Monk designed an experiment to strip out every variable except time. He kept subjects awake, fed them tiny identical liquid meals every hour instead of one big lunch, and tested their attention over and over throughout the day. If the early afternoon crash were purely about digestion, taking lunch out of the picture should have wiped it out. It didn't. People still dipped, right around 2 to 4 in the afternoon, and that dip lined up almost perfectly with a rise in something called the 12-hour harmonic of your body temperature rhythm, a term chronobiologists use for a secondary bump layered on top of your main circadian wave. Your master clock, the suprachiasmatic nucleus, sitting just above the spot where your optic nerves cross, doesn't run on one smooth 24-hour curve. There's a second, smaller wave riding on top of it, and that wave crests in early afternoon whether you've eaten a single bite or not. Scientists call this the post-lunch dip, which is honestly kind of a misleading name, because Monk's data and later work, including a 2011 driving simulator study where sleep-restricted subjects drove for 2 hours after either a 305-calorie lunch or a 922-calorie one, showed that the meal only tacks on a modest extra penalty to a dip that was already scheduled to happen. You were always going to get sleepy around 2 o'clock. Lunch isn't the cause. Lunch just has really unfortunate timing. But something else is going on too, and this part really is entirely about food. It starts in a tiny cluster of about 70,000 neurons tucked into your lateral hypothalamus, a region roughly the size of an almond that most people have never once heard of. These are called Orexin neurons, sometimes hypocretin neurons, and their whole job is keeping you awake, alert, and motivated to actually get up and do things. When they're firing, you feel switched on. When they go quiet, staying conscious becomes genuinely hard, and we know this isn't a guess because narcolepsy is essentially proof of it. In narcolepsy, those exact neurons get destroyed by an autoimmune attack, and people lose the ability to stay awake even when they're desperately trying to. In 2006, a researcher named Denis Burdakov, he was at the University of Manchester then, he's at Cambridge now, published something remarkable in the journal Neuron about these cells. It turns out they're directly shut down by glucose. Not through insulin, not through some roundabout hormone chain. Directly, Burdakov's team traced it to a class of ion channel called tandem-pore potassium channels, K2P channels for short, sitting right there on the surface of the orexin neuron. When glucose outside the cell climbs into the range you'd see after a meal, somewhere around 5 to 10 millimolar, those channels swing open, potassium leaks out, and the neuron goes quiet, essentially hyperpolarized into silence. And a follow-up study from that same lab in 2008 found something even stranger. The neuron doesn't even need to burn the glucose for fuel to sense it. Just having the sugar molecule sitting outside the cell is enough to flip the switch. So the exact neurons whose job is to keep you awake are, quite literally, wired to power down the moment your bloodstream fills up with the thing you just ate. And here's where it gets even stranger. The orexin system isn't just checking how much glucose is floating around, it's tracking the speed. Recent work out of ETH Zurich, published in Nature Neuroscience, came out of Burdakov's group after his move there, and they recorded from over 900 individual orexin neurons in mice as the animals went about their day. Most of those neurons weren't responding to the glucose level itself. They were tracking whether it was rising or falling. Some of them actually anticipated the peak of a glucose spike minutes before it arrived. So your arousal system isn't just reacting to how full of sugar your blood currently is. It's forecasting where that curve is headed and adjusting how awake you feel to match a prediction, which is a genuinely elegant bit of biological engineering. And it also means a big, fast digesting, high glycemic meal, the kind that sends blood sugar rocketing up quickly, should hit you with a sharper, faster drop in orexin firing than a meal that raises glucose slowly and gently. The physiology suggests white rice should knock you out harder than lentils would. Whether that specific comparison has actually been nailed down in humans, at the level of individual neurons, is a fair question to ask. The mouse recordings are direct observations, but the human side of this is inferred from blood sugar readings and behavior, not from watching human orexin cells fire in real time, because that's not something anyone can currently do in a living person. There's a second chemical pathway running alongside all this, and it was actually mapped out decades before Burdakov's work, back in 1971, by two researchers at MIT, John Fernstrom and Richard Wertman. This is the pathway the whole turkey myth was fumbling toward without really understanding it. Tryptophan is an amino acid, and it's the raw ingredient your brain uses to build serotonin, which then gets converted into melatonin, the hormone most closely tied to actually falling asleep. But tryptophan has a problem getting into your brain. To cross the blood-brain barrier, it has to use a transporter it shares with a handful of other, much more abundant amino acids: leucine, valine, isoleucine, phenylalanine, tyrosine. Under normal conditions, tryptophan basically loses that fight. It's outnumbered. What Fernstrom and Wertman figured out is what tips the balance: insulin. When you eat something carb-heavy, your pancreas releases insulin, and insulin pulls those competing amino acids, but not tryptophan, which happens to be bound to a plasma protein that shields it, out of the bloodstream and into your muscles. The competition thins out. Suddenly tryptophan has a much clearer shot across that transporter. Brain tryptophan levels go up, and within the hour, serotonin synthesis measurably increases. That's why a carb-heavy plate, pasta, white bread, a big serving of stuffing, produces more of this effect than a purely protein-based meal, even one that technically contains more tryptophan overall. It was never really about how much tryptophan was sitting on your plate. It was about how much insulin your plate triggered, because insulin is what clears the competition out of tryptophan's way. Now let's talk about how confident we should actually be here, because these two chemical pathways don't sit at the same level of certainty. The orexin-glucose mechanism is about as solid as central nervous system physiology gets. The ion channel is identified, the concentration range is mapped, and the narcolepsy connection makes the behavioral consequence undeniable. The insulin-tryptophan-serotonin pathway is also well replicated at the level of blood chemistry and animal studies. But that final step, that the resulting rise in brain serotonin is a major direct cause of feeling sleepy after a meal in humans, rather than just one contributing piece among several, is genuinely harder to pin down experimentally than the direct neuron recordings you just heard about. Serotonin itself isn't some simple sleep switch. It does a lot of different things depending on which of at least 14 receptor types it happens to bind. Here, it's best understood as upstream fuel feeding into melatonin production and general nervous system quieting, not as a light switch someone's flipping. But there's a third system, and this one doesn't live in your brain at all. It lives in your gut, and until fairly recently, nobody could really study it directly, because there was no good way to track exactly how much an animal had eaten, and whether it fell asleep right afterward, in the same experiment at the same time. In 2016, a team led by Keith Murphy and William Jha at Scripps Research, working with Alex Keene's lab at Florida Atlantic University, finally cracked this for fruit flies. They built something called the Activity Recording Cafe, a setup using machine vision to track both feeding through a tiny capillary tube and stillness in the same fly at the same moment. It ran in the journal eLife. And what they found was a sharp, short-lived spike in sleep immediately after a meal. But crucially, that after-meal sleep tracked specifically with how much protein and salt the fly had eaten, not sugar. A fly that just drank straight sucrose didn't get sleepy the same way. A fly that ate protein did. When the researchers silenced a specific set of neurons that carry the leukocynin receptor, that protein-triggered sleepiness disappeared entirely, while the rest of the fly's sleep stayed completely normal. This is a gut-to-brain signal, carried by peptide hormones, that's specific to what was actually eaten, separate from general fullness, separate from blood sugar. It's a fly, not a person, and that caveat matters. Murphy's team was careful about that themselves. But leukocynin-like signaling, and the receptor systems behind it, are deeply conserved all the way up through vertebrates, which is exactly why a result from an insect brain is worth paying attention to here at all. In mammals, the equivalent signal probably runs through a hormone called cholecystokinin, released by cells lining your duodenum the moment fat and protein arrive there from your stomach. Cholecystokinin acts locally on receptors sitting on the endings of your vagus nerve, the same nerve that runs from your gut, up through your diaphragm, through your chest, and into your brainstem. And those vagal fibers end in a structure called the nucleus tractus solitarius, deep in the medulla. From there, the signal doesn't just stop. It pushes onward into the parabrachial nucleus, and from there into the hypothalamus and the limbic forebrain, regions that have direct say over whether you're awake or asleep. Your gut isn't just quietly reporting fullness up to your brain like a fuel gauge ticking down. It's sending a hormone-coded, nerve-carried message all the way up a specific anatomical route, and that route ends exactly where your brain decides whether to keep you alert. You've probably heard a fourth explanation too, and honestly, it's the one that sounds the most intuitive, that blood gets diverted to your gut to help digest everything, starving your brain of oxygen and making you groggy. And here I have to be straight with you. This one's mostly a myth. Yes, blood flow to your stomach and intestines does go up after a big meal, sometimes by 30 to 60%, depending on how much you ate. But your brain doesn't just quietly lose out in that competition. Cerebral blood flow is tightly regulated on its own. The brain protects its own supply within a narrow range precisely because it can't afford fluctuation, and multiple studies using transcranial, Doppler, and other blood flow measurements have failed to find anything close to the dramatic drop in brain blood flow that would actually be needed to starve your neurons of oxygen or glucose. The blood diversion story is appealing because it's simple and easy to picture. It just doesn't hold up when you actually go look at the numbers. So here's what's really converging on you at around 1:30 in the afternoon, assuming you had lunch around 12:30. Your circadian clock's 12-hour rhythm is independently nudging your alertness downward on a schedule that would run whether or not you'd eaten a thing. On top of that, your blood glucose is climbing, and the Orexin neurons responsible for keeping you feeling awake and driven are being directly electrically silenced by that exact molecule through a potassium channel that wasn't even discovered until less than 20 years ago. On top of that, if your meal was carb-heavy, insulin is quietly clearing tryptophan's competitors out of your bloodstream, letting more of it slip into your brain to become serotonin and eventually melatonin. And on top of all three of those, if your meal had protein and fat in it, cholecystokinin is climbing up your vagus nerve from your duodenum to your brainstem, carrying a chemically specific message that has nothing to do with glucose, insulin, or your circadian clock at all. 4 systems, 3 of them running on completely independent clocks, completely independent chemistry. These aren't competing explanations trying to be the one true answer. They're 4 separate mechanisms that just happen to land on you at the same hour, because that's when you eat. There's something worth sitting with here. It took until 2006 to find the ion channel behind just one of these mechanisms, and until 2016 to build an apparatus sensitive enough to catch the gut-brain link in an insect that weighs about 1 milligram. This is one of the most universal human experiences there is. Nearly everyone who's ever eaten a big meal has felt some version of this pull toward stillness. And the actual wiring behind it was still being mapped out within the last 2 decades, in labs in Manchester and Zurich on a research campus in Jupiter, Florida. The feeling is ancient. The explanation is barely old enough to vote. So next time it happens, that heaviness behind your eyes 20 minutes after lunch, that sentence you were halfway through that suddenly takes real effort to finish, you'll know it isn't weakness. And it isn't the turkey. It's a potassium channel closing in a cluster of neurons about the size of a lentil. It's a hormone climbing a nerve from your intestine up to your brainstem. It's a 12-hour rhythm in a clock sitting above your eyes that was going to dip whether you'd eaten or not. 4 systems, 4 timescales, 1 afternoon. Your body was never betraying you at the lunch table. It was just doing 4 things at once, the way it always does. You only notice this particular collision because, for a few minutes, it wins.